So we do have almost a full 30 minutes left, which is what we were aiming for. So I'd like to thank the speakers for staying to schedule. As you ask questions or generate discussion, you can either address it to the panel as a whole or to, based on their talks, to individual members of the panel. And with that, I think we're ready to get started. So any questions or discussion from you all?
Use the microphones, please.
Yeah.
Yes. So two questions. Any information, maybe Andreas, any information on the combinations of beta-lactams, the carbapenems with on combination therapy? And from Sequoia, any information on combination of sotagliflozin with the other Sequoia drug? So two different questions.
Twanda, I'll pass that to David about the combinations. So usually we are testing the combinations of compounds in vivo. That's what we want, right? But in the case of beta-lactams, testing activity of those compounds in vivo, it is extremely challenging because it is very difficult to have the 60% time over the MIC that you are aiming for because of dihydropeptidase. So we have developed a new model that is a DHP1 humanized model to test the beta-lactams in vivo. So we are precisely doing this, but we do have extensive information about how beta-lactams behave when you combine them in vitro, and they are excellent companion drugs. So they exert the activity of any compound we have tested, and very particularly in the case of rifamycins. And about sotagliflozin,
So you asked me a question about combinations of sotagliflozin and other anti-TB drugs. As most of you are aware, sotagliflozin has a major active metabolite, and both the parent and the metabolite account for the majority of the exposure. In my opinion, that once we have looked at the potential for drug-drug interactions, that the combination of sotagliflozin with rifampin would make a very good combination. And that sotagliflozin is unlikely to be a perpetrator of an important drug-drug interaction. It may be the victim of an important drug-drug interaction, which remains to be determined. But based on what's known about linezolid, we feel that sotagliflozin in combination with other anti-tubercular, anti-TB drugs would be a good combination. We don't see any antagonism. We only see synergy and additive effects when sotagliflozin is combined with other TB drugs. So we're very much looking forward to clinical trials of sotagliflozin in combination with other TB drugs.
Just a clarification. I'm sorry. The only, in principle, what you said is entirely correct. But the caveat is that the metabolite shows a potential safety profile rather similar to linezolid in that its ability to interfere with mitochondrial protein synthesis is really very similar to linezolid. And if its metabolism were enhanced, the difference between it and linezolid would be reduced.
And I completely agree with that. However, when you look at exposure of the major metabolite in linezolid, the systemic exposure to the major metabolite of sotagliflozin is lower than linezolid, and we have been looking at the toxicokinetics in terms of the correlation of the trough exposure or the Cmin to systemic toxicity and inhibition of mitochondrial protein synthesis. So you and I are in agreement that we need to understand about conversion from sotagliflozin to the metabolite, and if that alters exposure.
Yeah, it all depends on how much you have to give.
Hi, Grania Brigden, MSF Access Campaign. I've got a question and a comment. My question is to Stephen, to ID about, and a follow on a bit on my question to Crispin is, with these regimen trials you're doing, is there any pediatric plans for these trials, and sort of how do you see that happening?
I have it, so I'll go first. Thanks, Grania. So currently there are no pediatric patients in stream, and in terms of stage 2, there are discussions about how it could be introduced, but there are no immediate plans.
And so for the TB Alliance regimens, we are committed to moving to pediatrics as quickly as we possibly can. PA824 in particular has a toxicity issue that has slowed pediatric development down. And so concrete plans at the moment, we're still waiting for enough data to allow us to do that. So the answer is no at the moment. There aren't concrete plans, but we are looking to move it as quickly as possible.
Thanks. And my comment is just to those with drugs earlier in development that I hope that there will be an open collaborative approach to bringing these drugs forward as regimens, complete regimens, rather than the sort of current adding it onto an optimized background regimen, which may not be the most effective or useful way. Thanks.
May I take the prerogative of a panelist and make a comment, an observation? Grania's question just made me wonder why we don't have a couple of hours dedicated to pediatric drug development at CPTR. Perhaps we should. Just a thought.
Thank you, Grania. Thank you, Crispin. Other questions or comments?
Jennifer Gardner from the Gates Foundation. Question to the group. I'm wondering if any of the clinical studies are actively looking at systemic markers, trying to even explore potential for a bacterial load measure in blood in patients.
So I'll speak for the TB Alliance studies. We do not have any target biomarkers right now, so we don't have any of those built into our studies. But you'll hear later about a biobanking initiative that we're part of, and we obviously support doing whatever work we can. And we've been open to actually using the platform of the STAND Phase 3 study and other studies to incorporate any work in this area. We just haven't found an appropriate or a well enough advanced program to actually do the work. And so we're looking. If anyone has any ideas, let us know.
Perhaps just a small comment as one that does clinical trials. It's in our 2-week studies, we actually often do that. We look at cytokines in blood and collaborate with immunologists and other interested parties and submit material to them that they analyze. But then the problem there is that you don't really have something to compare it to, because all you have is a dropping CFU count or an increasing time to positivity or something else that is a surrogate for what you actually want to measure. So if you want to do a biomarker trial with outcomes, you need a phase 3 study, and that is very, very difficult to organize and very expensive. But if you have such a compound, anyone has, we can gladly incorporate that into short-term trials. The Alliance has always been very helpful in incorporating that into protocols, and we're certainly happy to take an additional sample of blood from patients and make that available.
Yes, hi. Wim van der Velde from TBCAP. It's a follow-on question for Sequela. You mentioned that you will publish or that you will submit in Russia by the end of the year for conditional marketing approval based on the phase 2 data with studies with bedaquiline and delaminate, I understand. When will the data be public, and will you publish it in a peer review journal?
So actually the study is looking at optimized background regimen in MDR-TB patients with and without SQ109. So I'm not in a good position to actually say when the data will be publicly available or published. Right now, the analysis is ongoing with the goal of submission of the NDA for marketing and distribution in Russia only. Not in specific combination with bedaquiline and dolamitid. It would have depended on what was included in the optimized background regimen in the study. I thought I heard you say with bedaquiline and dolamitid.
Yes, I thought I heard you say that as well.
No, I'm sorry if I said OBR, so optimized background regimen.
To jump back to the previous question about the systemic biomarkers of bacterial load, there was a very interesting paper that was published recently in Lancet that came from an international consortium based more or less in South Africa that identified a small panel of genes, or two small panels of genes, that could predict your tuberculosis risk with a reasonable degree of accuracy, at least 6 to 12 months before the occurrence of tuberculosis. And this has many properties that makes you think it might be a biomarker for TB cure, not just risk. So we actually will be studying this in our multi-arm study of host-directed TB therapies that's funded by the Gates Foundation. We hope to get this up and running within the next few months.
Thanks. Dan Hartman from the Gates Foundation. Two questions. Since these compounds aren't likely to be pharmaceutical blockbusters, it seems like from a planning perspective you would want to focus on having the greatest impact in the shortest period of time. And so I'd love to hear folks comment on what they think the most efficient development pathway is to having the greatest impact. And two, for some of the compounds that were recently approved, it seems like my impression, could be wrong, it seems like the uptake of those are slow. And I wonder if that's meeting the expectations of when they were launched. And are there other enabling activities that could go on to help get those drugs to patients who need them?
We can go down the panel, I guess.
Let me start with the second question first, about what I call absorptive capacity of the field for new tools, including drugs. So in my working in TB, I've found us, the TB community, to be rather slow in adopting new technologies or new tools. And somehow, people may have come from hepatitis C or HIV, but once they come to TB, they catch that bug. And the slowness is sometimes just astounding. And it is a multifactorial situation. The field itself is, I think, poorly developed to be able to absorb these new technologies in a manner that is expeditious, that addresses the patient's needs immediately. And the National TB Control Program, that setup of the National TB Control Program, to me, appears to be one of the hurdles in getting things moving forward. My sense is if there existed the National Hepatitis C Control Program in countries, you probably would run into the same kind of bureaucracy that you run when you try to put a TB drug on the market. And sometimes the biggest challenge I've seen working from a pharmaceutical company perspective is the non-alignment between the National TB Control Program and the drug regulator, because we have to go to the drug regulator to get the registration. And sometimes we even find ourselves being pushed back by the National TB Control Program because their needs haven't been addressed. But it's not up to me to make sure that the National TB Program needs are being addressed. They should be talking to the drug regulator. They report to the same Minister of Health. So I think there's still channels that need to be, dots that need to be connected to allow us to get these drugs out there quickly and indeed reach more patients who are still waiting. The other question was about. Remind.
Trying to think through what the most efficient path from a drug
Efficiency.
development perspective
Efficiency to
is
drug
to
development.
get a product out to the people, most of the people who could use it, which I would say includes drug sensitive.
Correct. Yeah. You know, my sense if we didn't have CPTR, probably we wouldn't have made the progress we have made thus far. And I think these types of forum allow us to engage with people, to know who is doing what. And I see sort of a whole gamut of representation in this room, and it affords us conversations. I was alluding to the need to do a delaminate bedaquiline study, which was in fact first brought up in a meeting like this one in a hotel not far from here. And without that push, I think we wouldn't have actually gotten where we are. We aren't as far as we should, but nonetheless, we probably wouldn't have been this far. So I think forums like this and the level of collaboration and openness, you know, we tend to be rather closed when it comes to collaboration. Pharmaceutical companies are rather siloed entities. And I think if there's a framework to allow pharmaceutical companies to talk to one another in a manner that allows collaboration. TB is treated with regimens. You don't use a single drug to treat TB. So you can't really be in a framework of thinking about your drug. You should be thinking larger, and you should be thinking about how do we enhance collaboration to create the perfect TB regimen. So I think forums such as this, you know, the work that the Gates Foundation has done to allow us to begin to talk to one another when we normally wouldn't, is indeed part of what we need to be doing. If others have other ideas, here's a mic.
Yeah, I would like to comment that probably that situation might change. And I think one example, Carl mentioned it before, the TB drug accelerator, where all the companies, well, not all, but several big pharma companies, Merck, Merck, Lilly, Eisai, Sanofi, and some others, are all of us developing a new combination regimen all together. So I don't think we can afford being a zero, because the best development is to have a new regimen, not add our drug to an optimized background regimen. So we need a few of us to be successful in order to develop that regimen. Otherwise, it will be a waste. So that situation needs to be changed, and I think the bases are there. We just have to make it happen.
If I have my panacea hat on, I would like to say that we believe in a parallel development in drug-sensitive and drug-resistant TB, because we would not like to end up with a single drug with a conditional approval. Yeah, I think, like we have learned in the past, I think that is definitely hampering the wide distribution of drugs. So evaluate novel drugs both in drug sensitive and in drug resistance. So because if the safety profile allows, and I think that's our strategy, that we have a parallel development with an alternative backbone, or two alternative backbones, one based on the existing standard regimen, improving that, and the other backbone with novel drugs, like basically you're establishing now with NIX-TB and so on, and you could easily add a fourth drug or so, yeah.
Can I just add into this as well? I mean, I think the work that the TB Drug Accelerator is doing in the preclinical space is a good start, and I think we need to continue to build on that. So there is being some work done with MSF and a number of other organisations in this room about taking that collaborative regimen development into the clinical space and incentivising and changing how we incentivise TB R&D, so we're not depending on an end market. Because although we know there's lots of patients with TB, they may not necessarily be a pharmaceutical market in terms of what the market can bear for cost. So the 3P project is working on sort of looking at innovative models of financing, which ensures that the collaborative spirit that's in the preclinical arena is maintained into the clinical space, and that we're working and promoting regimen development, not single compound, and rewarding that appropriately. If anyone wants more information, please come and contact me about it.
We have a few more minutes.
Norbert Heinrich from Munich. I'd like to ask a question with regards to resistance development, and maybe to add to the last question. We have the aim of registering new drugs and regimens and bringing them out to the people as soon as we can, but then looking at maybe 10 or 20 more years in the future, another important aim to achieve is that these regimens might still be in use without widespread resistance having emerged. So I'd like to ask the question on which strategies would you envision to prevent the development of resistance against new drugs and or regimens?
Any takers?
I'll take the leap of faith here. So, appropriate use of antibiotics is probably the most important aspect in preventing resistance development. Back in 1959, when Joshua Lederberg, I think it was his name, the guy who discovered that bacteria can transfer genetic material from one to another, he said the battle between microbe and human may be obviously taking place, but the conclusion has not been determined yet as to who's going to win. So it could be the microbe, it could be the human, in terms of the use of antibiotics and the development of resistance. So I think, to me, education of the end user, both the patient and the prescribing physician, is one of the most important aspects. So sometimes we focus so much on the MIC or the drug exposure, but we ignore that these things will be used by human beings. And human beings have a tendency to gloss over PK and to gloss over MIC. And if we ignore that aspect of it, we will indeed put these things into a broken system, and they too will be broken. So I think we have to think of a complete spectrum of what needs to be done in order to make sure that we put these drugs in the right environment, that they'll be used appropriately. And that's how I think about it. But there's things we can do upstream to ensure that the human beings will not break these things. But I don't think I'm very well qualified to talk about those. So I'll pass on the mic.
So I think we have time for one last question. Okay, well, two last questions.
So it occurs to me on this particular question and the other questions that have been asked as probably one of the more fertile places for collaboration, because we do know in preclinical models and elsewhere, we do have the tools to design the new regimens going forward in such a way that we minimize the emergence of resistance. It seems to me the goal has been shortening of therapy, which I'm all for, but nobody has been developing doses for all these combinations we are talking about, or even collaborating on it, to forestall the emergence of resistance. Because we'll be here again in 10. It won't even be 20, right? So there is an opportunity for collaboration, but there are tools to do that. That's my point. And they are not being used going forward. We're doing things the same way we did for decades, and we'll get to the same place we got over the decades.
Thanks. If I may, I would also add, Crispin, that another area we've been very slow to implement at broad scale is knowledge of drug susceptibility patterns. We're still treating too many patients without knowing the drug resistance profile of the bug. We will first look for INH and rifampin resistance, declare it MDR, but be ignorant about what other drugs are reasonably used. And we are not yet fully implementing a comprehensive drug susceptibility pattern for individuals with TB, especially in the settings that are driving resistance. You don't see it in China, you don't see it in India, and I mean, I'm working with a student who just came back from a province in China where they're still diagnosing TB on the basis of AFB smear microscopy as of 2015. This is unacceptable. And I think that, I would call it, when asked about facilitation, a body that plays a key role is WHO, because they provide the normative policies that drive practices. And very often this ossified stance that you were describing in national TV programs have a lot to do with this is the way we've been taught to do it, and unless I get permission and policy approval from elsewhere, I'm not moving forward for fear of the unintended consequence. And we're too risk averse as a community, and we need to get out of that. I fully agree with your observations in that sense, and I've been part of this community for the last 2.5 decades, so I'm part of that problem.
Thanks, Ken. So we have one very last question, and
just to add to that, I mean, we have been listening to very interesting talk about drugs. Nobody has mentioned DST. So once a drug is brought to a market, we need to have a reliable way to test those drugs. And we have already had the experience with bedaquiline and delamanid that were brought to a market without very implementable tests. And this mistake should not be repeated in the future.
okay, with that, I'd like to thank the panelists for excellent presentations and discussion, and I'd like to thank the audience for your participation. Thank you very much.